So I would start the introduction. So before I introduce our speaker today, I would just remind you to visit our website, Connect Science. So we have all the previous talks recorded at this website if you are interested. And also, if you have questions for Dr. Chen today, please use the Q&A box instead of the chat box to type in your questions. So I will read out your questions at the end of the talk. So it's a great pleasure to introduce our speaker today, Dr. Junjie Chen from MD Anderson Cancer Center. So many of you may have known Junjie since his days at Dana Farber. Junjie finished his undergraduate training in Fudan University in Shanghai, China, and his PhD training at University of Vermont. And then he came to Boston and did a postdoc with Dr. Livingston at Dana Farber. In 1999, Junjie started his independent research position first at Mayo Clinic. He then moved to Yale University in a few years and then eventually ended up at MD Anderson. So he's now the professor and chair of Department of Experimental Radiation Oncology at MD Anderson Cancer Center. Also, Pamela and Wayne Garrison Distinguished Chair in Cancer Research. So Junjie has been a leader in the DNA repair field for many years. So his lab has identified and characterized a number of important DNA repair proteins, regulators of BRCA1 and ATR, ATM checkpoint pathways. So in recent years, his lab has also done important work on the Fanconi anemia pathway, DNA repair pathway. And more recently, in the last few years, Junjie's lab has done important work on synthetic lethality in cancer cells, helped to identify new vulnerabilities in cancer cells and develop targeted cancer therapy. So without further ado, Junjie, please go ahead. Welcome to the seminar series.
Sure. Thanks, Lee, for inviting me, and thank Claudia for organizing this. I mean, it's a great pleasure for me to be here, even so it's virtually. So as Lee introduced, I trained at Harvard, initially at Brigham and Women's with Ninja Data, and then I went on, did my second training with David Livingstone at the Dana Farber Cancer Institute. So I know a lot of people at the Boston area. It's unfortunate we cannot visit Boston these days, but it's nice to at least visit in virtual. So as Lee mentioned, we got interested in this DNA damage response pathway really because of my post training with David Livingstone. So in David's lab, we're interested in this the tumor suppressor genes BRCA1, which as many of you know, this is a critical tumor suppressor genes in breast cancer, familial breast cancer. So at the time, I worked very closely with Ralph Scully, one of your colleagues, and Ralph was able to show that this BRCA1 actually forms this very juicy or bright nuclear foci, which is the indication of maybe BRCA1 has a particular function in the nuclei. So this turned out to be true. And many labs, including our lab, Livingstone's lab, and Ralph's lab, and many others, has been able to follow up and really demonstrate BRCA1 is this foci turn out to be sites of DNA damage. And BRCA1 localized to these foci and also function in DNA damage response and DNA repair. In particular, BRCA1 is required for a checkpoint activation following DNA damage and involved in the homologous recombination repair, which is one of the major repair pathway following double-strand break. So as a protein, BRCA1 is a relatively large protein and contains N-terminal ring domains, which has been shown by many groups to have an E3 ligase and function and can modulate a lot of substrate in either polyubiquination or monoubiquination manners. The key substrate, essential BRCA function is still under debate, but we know a lot about BRCA1 through its C-terminal domain. so-called BRCA1 C-terminal domain. And now, probably many years ago, almost 18 years ago, our lab and Michael Yaffe's group be able to show that BRCT domain turned out to be a phosphoprotein binding motif. And so this really is suggesting BRCA1 uses this motif to interact with a lot of its binding partner in a phosphorylation-dependent manner. And this phosphorylation can be regulated in a cell cycle-dependent manner or damage-dependent manner. That's how BRCA1 may shift partners and interact with different proteins at different time to carry out its functions. So through the work of many groups in the field, and we now know a lot more about how BRCA1 interact with a lot of other DNA damage repair or response proteins. So we know that BRCA1 is actually recruited to the site of the break, as I show you in the earlier slides. Localizes foci structure really depend on very complex pathway which are initiated by H2X phosphorylation, which is a damage-induced phosphorylation event, which lead to the subsequent recruitment of MDC1 and then to recruitment of two downstream E3 ligases, RF8 and RF168, lead to the formation of polyubiquitination chains, which is then recognized by a large protein complexes. On one hand, can hook onto these polyubiquitination chains. On the other hand, contains a phosphorylation site, can be used by BRCA1 C-terminal region to bind and recruit BRCA1 to the DNA damage site. So we know a lot about how this regulation of this damage-induced cellular event and how does that BRCA1 can be recruited. We also know a lot about other BRCA1 interacting protein, including CTIP, which is a partner of mRNA complex, which is involving the detection of double-strand break and involving the early end resection step. So this BRCA1 interact with this complex important for the initiation of DNA repair. So BRCA1 also interact with BAHA1, which was cloned by Sharon Cantor when she was in Livingstone's lab. And this interaction appears also interact with TOPO-BP1 and important for the unwinding and extension of the single-strand regions. So BRCA1 also interact with BRCA2. So when I was in Dave's lab, we showed that BRCA1 interaction with BRCA2, and Dave's lab also cloned another protein called PAB2 by Binsha, and PAB2 turned out to be a bridging protein between BRCA1 and BRCA2. And BRCA2 bind directly to a recombinase called REFLIP1, and this is critically important for the loading of REFLIP1 and starting homologous recombination and homologous search. So you can see BRCA1 interact with a number of proteins and are known to be involved in DNA repair and checkpoint regulation. So we also know since then a lot about not only BRCA1 function and how to take advantage of BRCA1 deficiency. So I'm sure this audience are very familiar with the story of PARP inhibitors, which has been really demonstrated to be very effective in treating both BRCA1, BRCA2 deficient tumors. And this has been really rapidly translated into the clinic, has now been used for breast, ovarian, and other cancers with BRCA mutations. So we also know that this, at least for BRCA1, the sensitivity to PARP inhibitor is actually depend on another DNA repair pathway or DNA repair gene called 53BP1. So there seems to be a competition between these two repair pathway in terms of how they're repaired, and the outcome determines the PARP sensitivity. So this has been studied probably in the last 10 years very extensively by many groups, so-called double-strand break pathway choice, really trying to understand how this pathway choice are made and how this pathway choice determine the outcome for PARP resistant or sensitivity for BRCA1 deficient cells. So today I'm not going to talk about anything about BRCA1, but just using BRCA1 as an introduction, really to tell you a little bit more updated story we're interested in, really in the field, trying to understand additional replication and the DNA damage checkpoint and how to take advantage of this knowledge to really develop a better anti-cancer therapies. So besides working on BRCA1 or other things, our lab is also interested in replication checkpoint control for quite a few years. As you probably heard from Li Zhou and Steve Elich's group, they're one of the initial group really demonstrate many of a DNA damage event, as long as they can cause replication or single-strand DNA. So they will be initiated by, this will lead to a single-strand DNA RPA complex. And this probably is the signal which eventually activated the ATR check1 pathway. So this is a critical pathway in the cell because a number of not only damage events, but also oncogenic stress because of this uncontrolled replication can also lead to the activation of ATR check1 pathway. So we've been interested for quite a few years, mainly because we study a protein called TOPO-BP1, which is a multiple BRCT domain containing protein. So we're interested in how TOPO-BP1 can integrate multiple phosphorylation and other signals lead to the activation of ATR check1 pathways. So several years ago, we also showed that RPA can also bind directly to another protein called ETA1. ETA1, like TOPO-BP1, can also bind directly to ATR and activate ATR check1 pathways. So as I mentioned, this ATR check1 pathway seems to be response to a lot of cellular damages and replication stresses. This turned out to be very critical for cells, especially tumor cells, to survive following a number of stresses. So therefore, a lot of companies are interested in develop various inhibitors of this pathway, including several inhibitors for ATR, several inhibitors for check1, and inhibitor for V1. So quite a few of these inhibitors has been moved on to phase 1 or phase 2 trials, really trying to take advantage of the known activation of ATR check1 pathway in tumors and to see whether cells with a certain defect will be extremely sensitive to ATR or check1 pathway inhibition. So as you probably heard from Li Zhou's group, Li's group has been shown that a number of different stress scenarios can lead to the hypersensitivity to ATR inhibition. So we're also interested in this particular pathway. It's really interesting, probably more broadly, to see whether we can really use in the recently developed for quite a few years, the CRISPR-Cas9 technology, to really allow us to understand how what kind of defect can lead to the cellular sensitivity to ATR inhibitions. So I'm sure all of you are very familiar with the CRISPR-Cas9 technology and the whole genome screens using different drugs. In this case, we're using the ATR inhibitor, this AstraZeneca compound. So I really have to highlight all this work made possible by our collaboration with Trevor Hart. So Trevor Hart is a bioinformatician and also experimental biologist. he trained with Jason Moffett in Toronto. And over there, when he was there, he really involved in the development of whole genome sgRNA libraries, this TKO Toronto knockout library. The version 3 is the one we used. And so he not only be able to really do the experiments to help us to establish how to do this experiment, but he's also really help us to do the data analysis, which I think is very critical for really understand the hit come out of this kind of large scale screens. So with his help, many people in my lab has to start to really using this whole genome screen to answer a number of questions in DNA damage or cancer field. So today I'm going to highlight the several studies done by Cao Wang and Xu Fan in my lab. So as a screen, it's pretty straightforward. You take the cells, you infect it with this whole genome library, and you treat the cells with either control or any drug. In this case, is the ATR inhibitor. You follow the cells for a certain doubling time and you sequence them. So what you can see is, so this is the T0, this is after infection, and you can see after the 20 doubling time, you can see there's dramatic change of a GRN distribution. And most of them are due to cell-specific or common essentiality genes. So really, we're looking for a small difference between the ATR inhibitor treated cells versus the DMSO treated cells. I think the powerful method developed by Trevor Hart really allow us to tell the difference between these two and really derive meaningful information from this type of screens. So initially, when Cao started this project, we did this synthetic lethality screen with ATR inhibitor in 3 different cell lines. And you can see that I using the circle to circle out either the top sensitive genes or top resistant genes. You can see there's an amazingly similarity between 3 cell lines. You can see RSH2A and B show up as the top genes in all 3 cell lines. Really highlight the powerful of this technology. And on the other side, you can also see the resistant to ATR inhibition, CDC25A and B, consistent come out in all 3 settings, cell lines. Again, really suggesting combined with careful experimental approach and a bioinformatic analysis, we'll be able to really derive core essential genes which are required for the sensitization or resistance to ATR inhibitors. So this the list here is just to show you the GOI analysis for all the process which when deficient sensitizing cells to ATR inhibitions. As you can imagine, this including everything we know about involving DNA replication and DNA repair, including double-strand break repair, intra-strand break repair, or others. So really saying this analysis will be able to not only help us to understand how cells become sensitive to ATR inhibition, but also understand how this pathway may be important involving the cellular response to different types of stresses. So some of this study has already been published, so I'm not going to go into detail, but really just give you a summary what we learned from this initial screen with ATR inhibitors. So as I mentioned, one of the major hit from this screen is the RSH2. So RSH2 is a heterotrimeric complex which is involving removing the RNA primers during replication and also remove the missing cooperative ribonucleotide. So this is known to be important for DNA replication involving replication. The reason we're interested not only because all of them are identified in our screen using 3 different cell lines, but also because RSH2 is also frequently downregulated in prostate cancer shown by other groups. So we be able to follow up and using the PDX models available from MD Anderson, been really able to demonstrate that indeed a subset of PDX models contains RSH2, especially RSH2B deficiency or loss, which really is suggesting this can be potentially a biomarker for the ATR inhibitors. Indeed, we showed that RSH2 deficient tumor cells are sensitive to ATR inhibition, both in vitro and in vivo using xenograft models. Again, validating that RSH2 loss may be used potentially as a biomarker for the cellular sensitivity to ATR-based therapies. So I want to mention that RSH2 was also discovered by Dan DeRoche's group with their screen of PARP inhibitors. So it turned out to be deficiency in RSH2, not only sensitizing cells to ATR inhibitor, but also to PARP inhibitors. So there's a number of, groups are interested in developing IHC or other assays for RSH2. Hopefully this can be used as a companion biomarker diagnosis trying to determine whether the patient can be sensitive to either PARP or ATR inhibition. So another things we really derive from this type of screen is we can also identify new genes involved in DNA damage or replication stress, because seems to be any deficiency in this process make the cells very sensitive to ATR inhibition. So this led us to identify at that time unknown gene called the C17OFF53, which is important for cell survival following ATR inhibition. And these cells are extremely sensitive to intrastrand cross-linking. We'll be able to show it's the DNA binding protein interact with RPA and act with the MCM8-9 involving probably intrastrand cross-linking repairs. So both Dan's group and Abato's group independently discovered the gene and shown its involvement in the intrastrand cross-linking repair. So this kind of analysis really highlight when we perform these screens, we need to probably potentially not only isolating some kind of biomarker may be important for the future clinical studies, but also really identify new genes may be involved in different type of DNA damage repair or replication processes. So with this success, Cao decided to really expand this study to really to do a more encompassed study contains different ATR, DDR kinases, which are being developed, trying to use them for the anti-cancer treatment. So this not only including ATR inhibitor, but also including ATM inhibitor, DNA PK inhibitor, as I mentioned, also checkpoint inhibitor. We also include mTOR as a control because ATR, ATM, DNA PK are all large PI3 kinase-like kinases. So we include an mTOR inhibitor as control and also include the PARP inhibitor as controls. So this study is done in mostly in 293A cells, but with different inhibitors, really trying to get into some commonality and differences between different inhibitors and to see how we can take advantage of this knowledge in the future clinical trials. So this is all the hit. We class them together. As you can see, as I mentioned, ATR inhibitor really shown clone's many cellular process, including replication and repair deficiency, show extreme sensitivity to ATR inhibitions. As you can see, this CHEK1 inhibitor will behave similar to ATR inhibitor. However, they only hit fewer genes compared with ATR inhibitor. This probably makes sense because CHEK1 believed to be functioning downstream of ATR. Probably some of the ATR regular process are not regulated by CHEK1. So if you look at the ATM, so you can see there is some overlapping with the ATM and the ATR. However, there's also a lot of distinctions. So ATM are not, a lot of defect in the cellular process may be sensitive to ATR inhibitor, but not to ATM inhibitor. And DNA PK, we get even fewer hits. And with the mTOR inhibitor, we get a totally different hits, and most of the hits are involving mTOR signaling. So this is just a highlight. These screens are successful, and we be able to really distinguish the mTOR inhibitor with all the other inhibitors, and also suggesting that this different inhibitor probably are very specific because they don't have an overlapping in the targeting the mTOR signaling pathways. So if we just analyze this data, we can find quite a few commonalities. So the most obvious things turn out to be is a Fanconi pathway, like Lee mentioned. Deficiency in Fanconi pathway make the cell sensitive to ATR inhibitor, ATM inhibitor, and all CHEK1 inhibitors. As I mentioned, that DNA PK seems to be only hit a few of this DNA repair process. And interestingly, the most of this hit shared with, some of them shared with ATM and ATR turn out to be involving BR pathway or single-strand break pathways. As I mentioned, the mTOR really totally lead to the identification of a different subset of genes involving the mTOR regulations. So we decided to drill down to trying to see whether we can identify some unique for each of these inhibitors, which really has not been highlighted in the previous studies. So when we look into CHEK1 data, we find that there are cells deficiency in this YWHAE, which is a 1433 epsilon, make the cells extremely sensitive to CHEK1 inhibition. So we also perform CHEK1 screen in a different cell line. In this case, the HCT116 cells. Again, we get this 1433 epsilon as one of the major hit to sensitizing cells to CHEK1 inhibitions. So I think this audience is probably very familiar with cell cycle regulations. And so 1433, as you all know, is has a forcible binding ability. And the number of 1433 in the cells, and they may have a overlapping or specific function in either cell cycle regulation or number of other events, which requires the phosphorylation-dependent regulations. So really trying to understand the specificity about this 1433 protein, we look at all the other 1433 protein genes in our library, and you can see that this 1433 epsilon in both cell lines is the only one really show very significant depletion following CHEK1 inhibition, suggesting that 1433 epsilon depletion may be the specific for this the cell sensitivity to CHEK1. So just to follow up, we'll be able to really just using sgRNA to knock out or knock down this 1433 epsilon and show these cells are now become very sensitive to CHEK1 inhibition. And in addition, so wild-type cells, if you treat with a checkpoint inhibitor for a long time, and you can start to see some S-phase defect, but pretty mild S-phase defect. However, in 1433 knockout cells, and you see a dramatic S-phase or G2 arrest or accumulation following checkpoint inhibition. So suggesting that this checkpoint and 1433 may both of them involve in the regulation of this S2G2 regulation, which you need both of them, otherwise the cell become very sensitive to checkpoint inhibition. Again, the reason we're interested in this 1433 epsilon is involved because this gene is located at 17p 13.3, which is a frequent microdeleted region in cancer. And there's a number of previous studies suggesting that silencing or loss of the 1433 can be observed in different cancers. So again, suggesting that maybe this loss of 1433 may make the cells very sensitive to checkpoint inhibition, which need to be further determined. So we also look at other sensitivity or resistance to other inhibitors. In this case, we actually look at the resistance to ATM inhibitions. As you can see, that the top gene shown when lost showed resistance to ATM inhibition turned out to be KLHL15. So KLHL15 is actually been studied previously, and this is from shown by Satori's group that this form a complex with choline 3 and involving the degradation of CTIP. As I mentioned earlier, CTIP is a BRCA1 interacting protein, also function together with the mRNA complex involving DNA end resections. So we're interested in how to see whether this indeed is true. We actually perform another screen using a higher dose of ATM inhibitors. Again, this KLH15 turned out to be on the top of the list, really suggesting loss of this KLH15 really lead to cells resistant to ATM inhibitor. So indeed, and we can knock out this KLH15 and these cells become resistant to ATM inhibitor. So we believe these are indeed through the CTIP, regulation of CTIP protein levels, and we're using Mary Jason's HR report assay to show that when you knock out KLH15 and the HR is efficiency increased, and this is totally dependent on CTIP, because if you deplete CTIP, then you totally abolish this increase and lead to reduction in the HR activities. So as I showed here and also repeating here, if you knock out KLH15, the cell becomes resistant to ATM inhibitors. And you can see exactly the same thing if we overexpress CTIP. So overexpression of CTIP also lead to the resistance to ATM inhibition. And again, if you knock out CTIP, the cells become very sensitive to ATM inhibition. So really suggesting that this regulation of a CTIP or CTIP level is very important to determine the cell sensitive or resistant to ATM inhibitors. So we're interested in to see whether really, as I mentioned, the number of drugs, including the PARP inhibitor, ATM, ATR, and DNA PK inhibitor has been now developed and go into phase 1, phase 2 trials. There's a lot of interest in trying to see whether combining these inhibitors will lead to a better tumor controls. So we're also interested in this question to see whether we can combine some of this inhibitor can lead to a different sensitization or different resistance mechanism, which are worth pursuing. So the initial experiment we did is we take the white cells. We also made POP1 and 2 double knockout cells. And surprisingly, this double knockout cells, POP1 and 2 double knockout cells are extremely sensitive to ATM inhibitors. And this is not the case for ATR inhibitor. You can see some sensitization of an ATR inhibitor, but not as dramatic as to the ATM inhibitors. And if you're using DNA PK inhibitor, we pretty much didn't see much changes between wild type and POP1 to double knockout cells. So this is directly looking at IC50. As I mentioned, that in the double knockout cells, you can see a dramatic change of IC50 to ATM inhibitor, suggesting these cells becoming extremely sensitive to ATM inhibitor. But it's modestly sensitive to ATR inhibitor, and we don't see much difference with DNA PK inhibitors. And as people really follow the POP literature, knows that a POP1 inhibitor, POP inhibitor, not only inhibit POP1 to catalytic activity, but also has a dominant negative activity functions, which are not going to be reflect on these double knockout cells. So with that, we also did a co-treatment. We treat with ATM inhibitor or POP inhibitor either alone or in combination. Again, the data are consistent, suggesting ATM and the POP inhibitor combination show the most synergistic effect in the tumor controls. So suggesting that this combination probably going to be useful, at least based on the in vitro study we performed here. So every story has a little bit twist. In this case, the twist is when we're using the ATM inhibitors to do a screen, as I mentioned, initially we focus on KOH-15 because which is the on the top of the list. We can see that even ATM show up in these screens. So what does that mean? So most time, if you have your target gene show up in the resistance screen, means the target may have a dominant, the inhibitor may have a dominant negative effect. So the dominant negative effect of ATRM inhibitor has been really documented previously by a couple of different groups. So we're interested in whether why normally we don't see it by treating the cells with ATM inhibitors. So we find that it's really depend on how long you treat the cells. So the dominant negative effect really need time to accumulate, probably due to this dominant negative lead to some sort of DNA damage, DNA break formation, which we were able to validate using the COMET assays. So what I show you here is in wild type cells treated with this low dose of ATM inhibitor, which as you can see, even after 24 hours, you don't see much change in the cell cycle distribution, cells don't die. However, if you treat it for longer and longer time, for 72 hours, you can now start to see a huge G2 accumulation and sub-G1 apoptotic cells, suggesting this is probably due to accumulation of damage, and this eventually lead to cell death. And this is totally depend on the presence of ATM protein. So in ATM, so we also made ATM knockout cells. As you can see, in ATM knockout cells, we can treat with the same concentration and for a long period of time, and you don't see any effect. And so this become interesting and puzzling to us. So we went back to really to do this, as I mentioned, in our hands, ATM inhibitor and a PARP inhibitor seem to be has a synergistic effect. And turn out to be this synergistic effect totally depend on this dominant negative effect of ATM inhibitors. So this is just showing you the data. If you just look at the blue line, which is the wild type cells, and this brown line, which is the ATM knockout cells, you can see when you treat with a PARP inhibitor, this case olaparib, there's almost indistinguishable. So you really in ATM knockout cells or wild type cells are not sensitive. The ATM knockout cells are not more sensitive to PARP inhibitor than the wild type cells. However, this is totally different. If you treat with ATM inhibitor, now you see a huge synergy between ATM inhibitor and a PARP inhibitor in this case. So I mentioned that we showed that the loss of this 1433 protein sensitizing cells to CHEK1 inhibition. We're following up this and trying to identify tumors or tumor samples which may have shown a loss of this 1433 and really direct testing whether these tumor cells will be sensitive to CHEK1 inhibition. And the screen will also uncover many different components of a BR or single-strand break pathway. The interesting aspect of this pathway is it depends on different component, and different component loss will lead to a different pattern of a sensitivity or resistance to ATR inhibitor, ATM inhibitor, DNA PK inhibitor, suggesting that BR pathway deficiency or maybe the intermediate step of this BR pathway deficiency really are important for their sensitivity to different type of inhibitors. So I spent quite some time really focused on KRHL15, suggesting their loss of this gene lead to enhanced CTIP expression, which maybe explains the sensitivity to ATM inhibition. And this ATM inhibition also have a very prominent dominant negative effect. And ATM and PARP1 show a synergistic effect in tumor controls. And this synergistic effect totally depends on the dominant negative effect of ATM inhibitors. So what this means to us or maybe to people really using these inhibitors. So this really is suggesting that PARP inhibitor may not be show any significant effect on the ATM deficient tumor cells. I believe this has been tried. I'm not sure the outcome of these clinical trials. And the other things is that the PARP inhibitor plus ATM inhibitor may have a combination, may have a dramatic synergistic effect on tumor control. Hopefully, somebody will be interested in test in the real clinic whether this indeed is the case. So with that, let me shift the gear and tell you a different story really done by Xu Fan in my lab. And she is also interested in this synthetic lethality screen, but he's interested in probably a more broad question, really to say, I mean, how can they be used for a common loss of tumor suppressor genes? I think this probably is the goal of a synthetic lethality, is to see whether the loss of any tumor suppressor gene will be, I mean, extremely sensitive to particular manipulations like a loss of some other additional genes or as a target. So with this goal, he decided to really make a lot of isogenic cell lines in HEC293A cells. I know there's a lot of effort has been done in Broad and other institutions, really screen for 100 cell line using this synthetic lethality approach or CRISPR-Cas9 approach, and which a lot of data available at the DeepMap. And however, different cell lines, tumor cell lines carry a lot of different mutations. So it's very difficult to compare the genetic and genetic interactions because there may be multiple mutations in these cell lines. We're not saying this is perfect, but this is what we tried. So what we tried is we select 12 common tumor suppressor genes. As you can see, this is based on their protein-protein interaction database. And you can see this suggesting these different tumor suppressor genes contains different functions, including remodeling, cell cycle regulation, or negative regulation of oncogene signals or stress responses. And you can see there's more interactions that the genes being well studied, like p53, and the genes only show in a subset of the tumors, like F1s, a little study, so there's very little interaction data available. So what she did is to really knock out each one of them one by one, generate so-called isogenic cell lines with a different knockout of these tumor suppressor genes, and really just trying to understand how this loss of a tumor suppressor gene in a common cell line, how they behave, whether they're going to lead to distinct vulnerabilities based on the CRISPR-Cas9 squeeze. So I think the first interesting thing after he made all these cell lines is interested in this CDH1, which is catenin. But catenin, you can see that it's not only lost in our own knockout cells, but loss of different tumor suppressor genes often lead to the loss of beta-catenin. So suggesting that this different tumor suppressor gene loss may also affect beta-catenin expression, may also affect EMT in different settings. And another interesting thing is most time we think loss of tumor suppressor gene will allow the cell to proliferate better. This turned out to not to be the case, at least in HEC293A cells. So the red line is the wild type cells, and the only one really show some growth advantage is the loss of PTEN, which negatively regulate PI3 kinase pathway. Loss of any other genes, including loss of PP3, actually make cells grow a little bit slower. So all these previous assumptions that loss of tumor suppressor gene will help the cell proliferation, probably not true, at least not true in the in vitro setting when we supplement the cells with 10% FBS or the nutrient which are available. So this is just to before we show you any data, just convince you the data are reliable. So this is UC curve position recall to really show off some really has a very, the screen data is very reliable, and we can cluster most of the wild type controls in different batch of experiment clustered together. And when we analyze all the data from these screens, you can see about 40 or 50% of them seem to be shared between different tumor suppressor genes, and another 40, 50% of them seem to be unique to a particular tumor suppressor gene in these particular cell settings. So what we can learn from this kind of screens. So one thing we learn is if we take all the shared hits together, seem to be all the shared hits are mainly involved in DNA damage and the metabolic pathways. And you can see this interesting correlates link and the number of nucleotide metabolism pathway or mitochondrial process come up as the top hit. We'd be able to validate DHOH loss seem to be sensitizing to both VHL and the BAP1 loss, and it's more sensitive to VHL loss, and we did to validate using 3 different VHL knockout cells to show that indeed these make the cells more sensitive to inhibitor of DHOH. So the other things we learned from this type of screen is there seems to be some synthetic lethality among different tumor suppressor genes. In this case, when we analyze data, we find that in LKB1 knockout cells, these cells are actually become sensitive to loss of TSC2. As you all know, TSC2 is actually another tumor suppressor gene involving mTOR pathways. So as you can see from the fold change, the GNA fold change here, TSC2 loss normally help the cell proliferations. You can see that this are actually enriched in Y-type cells and also enriched in P10 knockout cells. However, in LKB1 knockout cells, this TS2 GNA is actually depleted. So really suggesting that there seems to be some genetic interaction between the tumor suppressor genes, and we'll be able to validate in using the non-small cell lung cancer samples, because the LKB1 loss is pretty common in non-small cell lung cancers. And we can see there's a mutual exclusivity between these two. I mean, it is significant, but not very dramatic, because TSC2 loss is actually pretty rare even in non-small cell lung cancers. So to further show a different data set, we also be able to notice that in BAP1 knockout cells, KDM5C also seem to show a pretty strong genetic interaction with the BAP1 loss. So, and BAP1 is mostly being shown to be mutated or lost in the renal cell carcinoma. And we take I believe this is deep map data when this screen was done in renal cell carcinoma with BAP1 loss, and we compare our data set with their data set. The only thing overlapping between these two data sets is only one gene. This gene is KDM5C, and there's negative correlation, suggesting this loss are not tolerant in the cells. Indeed, so there's a lot of data available from the clear cell renal cell carcinoma, and the BAP1 loss is pretty common, and KDM5C is also quite common. And you can see there's a very dramatic mutual exclusivity between this loss of these two tumor suppressor genes, suggesting their function may be antagonist to each other. So with that, I just want to really thank the people who actually did work. So all this work really involving synthetic lethality screen really allow us to understand how gene-gene interaction and gene-drug interaction occurs, and help us really to find potential biomarkers, really be able to use this biomarker hopefully in the clinic in the future. Again, I want to really highlight our collaboration with Trevor Hart, and we managed to convince Trevor to move to MDS about 4 or 5 years ago. And because he's a presence in MDS, and also because he's really engaged in all this collaboration with us, allowed us to be able to perform these screens and allows to analyze the data. And again, so I want to thank Cao and Xu, who are the major driver behind this project. Thank you for your attention.
Thank you, Junjie, for your wonderful talk. Just a reminder to the audience, if you have questions, please type in the Q&A box, and I will read out your questions one by one. So just to get started, I will ask my first question. Yeah, so very interesting, the dominant effect of ATM inhibitor together with PARP inhibitor. So do you know if ATM inhibitor can trap ATM on DNA, or it's helping to trap PARP, perhaps?
We haven't tested directly. I mean, we tried to see whether there are more chromatin association of ATM after the ATM inhibitor. The result is not very convincing. So I don't know whether it directly trapped the ATM or has some function, maybe trap the others. I don't think it's trapped PARP. But we haven't done the triple knockout cells. Yeah, we don't know. So, but ATM inhibitor by itself has a dramatic effect. This whether this is due to trapping of RNA repair intermediate, whether it's a ATM physically get on it, we don't know.
Okay. All right, so there are a few questions in the Q&A box. So the first question is, so based on your data about the dominant negative effect, how can you explain the clinical, how can you explain that the clinical ATR inhibitors show activity in ATM deficient tumors?
ATM deficient tumors.
So
I don't think we compared the ATM deficient. So ATM deficient cell is still sensitive to ATR inhibitor. It's just not, it depends on what you compare to, right? If you compare to wild type, the cells, the ATM knockout cells are still sensitive to ATR inhibitor, but it's not as dramatic as a PARP inhibitor.
Mm-hmm. Okay. All right, so here's another one. Can you share your thoughts on why a basic size resolution or BER is synthetic lethal with DNA PK inhibitors?
We don't know. So, and I couldn't remember the details. I don't think it's all of them. So I believe the data show that, so we get almost all the BER pathway component in the ATR inhibitor screen, but from the ATM inhibitor screen or DNA PK inhibitor screen, we only get a few of them. I think Apex one may be the one showing very sensitive to DNA PK inhibitor. It shows sensitive to ATR inhibitor, but not to ATM inhibitors. So there's a lot of nuances about exactly which one you inhibitor loss sensitive to different inhibitors. We haven't go through mechanistically trying to dissect them out yet.
Mm-hmm. Okay. So this is the third question. So hi, a very amateur question. For double knockout of PARP1 in ATM, were these cells generated by knocking out PARP1 first and then introducing ATM lenti guide RNA, or introducing PARP1 and ATM guide RNA together in cells to generate double knockout? So yeah, what was the MOI double infection used? and were multiple guide RNA or just one guide RNA used for PARP1 to generate knockout cells?
Okay, so this is probably more details. We actually didn't make the PARP1, 2, and ATM triple knockout. So what we did make is a PARP1 knockout, and then we made a PARP2 knockout individually, and then we made a PARP1, 2 double knockout cells. So for most of the experiment, we used the double knockout cells because PARP inhibitor has been shown to inhibit both PARP1 and PARP2, probably not so much for PARP3. So that's why we mostly using PARP1, 2 double knockout cells. And for the ATM, additional ATM knockout, we just using GRNA. We didn't isolate any single clones.
Okay. So yeah, I have a second question actually about the tumor suppressor part. So it's interesting, most of the tumor suppressors, when you knock them out, cell proliferation actually slow down a little bit. But DNA repair pathway is still the common hit. Why is that? Yeah.
Yeah, so yeah, I don't know why is that. I mean, DNA repair is still common hit, and Fanconi pathway come out all the time. So we don't know. It's again, I mean, there's more and more studies being published, and Fanconi pathway seem to be a pop out in everybody's screen. So we don't know there's a truly. I mean, I don't want to say it's all artifact or screen, but that's come out pretty common. So we don't know how to explain that. And of course, there is a nucleotide, I mean, synthesis or this pathway also come up pretty common. So maybe that's really just reflecting these tumor suppressor genes. Maybe loss of it will indeed, I mean, has some stresses, but it really, it doesn't help the cell grow in the normal situation. So probably help the cell grow under some other stressful situation, which we don't know what it is because we're doing this in tissue culture settings. So I think for us, the goal in the future is really to move to probably more in vivo screen. Because I mean, I just don't think I mean, we're going to, if this tumor suppressor gene interaction in the tissue culture setting may only reflect part of the function of these tumor suppressor genes, not the whole function of tumor suppressor genes. So I think this need to be probably done based on in vivo studies.
Great. Yeah, I think that's all the questions we have now. And it's exactly 1 o'clock. It's perfectly on time. Yeah. Okay. Yeah. If no further questions, yeah. Well, Junjie, thank you very much for your wonderful presentation today. And I'm sure a lot of us learned a lot from your talk. Thank you. Thank you very much.